cell cycle. And for further information, as always, you can see the separate article on cell cycle in Wikipedia. So the cell cycle is a series of events that take place in a cell that cause it to divide into two daughter cells. These events include the duplication of its DNA and some of its organelles, and the subsequent partitioning of its cytoplasm into two daughter cells in a process called cell division. In eukaryotes, such as animals, plants, fungal, and protist cells, there are two distinct types of cell division: mitosis and meiosis. Mitosis is part of the cell cycle in which replicated chromosomes are separated into two new nuclei. Yeah, so this is two chromosomes, or this replicates chromosomes. They're separated into two new nuclei. Cell division gives rise to genetically identical cells in which the total number of chromosomes is maintained, and that is in this mitosis. In general, mitosis, which is a division of the nucleus, is preceded by the S stage of interphase, during which the DNA is replicated, and is often followed by telophase and cytokinesis. And this divides the cytoplasm, organelles, and cell membrane of one cell into two new cells containing roughly equal shares of these cellular components. The different stages of mitosis altogether define the mitotic phase of an animal cycle, the division of the mother cell into two genetically identical daughter cells. The cell cycle is a vital process by which a single-celled fertilized egg develops into a mature organism, as well as a process by which hair, skin, blood cells, and some internal organs are renewed. After cell division, each of the daughter cells begin the interphase of a new cycle, which is interphase again, during which DNA is replicated. And again, to recap, mitosis is the division of the nucleus. That's in which replicated chromosomes are separated into two new nuclei. So yeah, you're dividing the nuclei into two. And hence, you're dividing the nucleus into two nuclei. Now let's continue further. So just note, a chromosome, because remember this is your replicated chromosomes inside the nucleus is separated into two nuclei. The chromosome is a long DNA molecule with part or all of the genetic material of an organism. And I had gone over this earlier, but just a quick recap. And a chromatid, we're going to get to that in a bit, is one half of a duplicated chromosome during the synthesis S phase and occurring between gap 1 and gap 2 phases in the interphase portion of the cell cycle. So there's one half of a duplicated chromosome, and we'll see that in a bit. And a sister chromatid refers to the identical copies of chromatids formed by the DNA replication of a chromosome, with both copies joined together by a common centromere. So if you have here, the centromere is a specialized DNA sequence of a chromosome that links a pair of sister chromatids called a dyad. All right, continue further here. So here's an illustration here. So sister chromosomes in separate cells. The paternal, actually no, that's at the bottom here. So the paternal blue chromosome and the maternal pink chromosome are homologous chromosomes. So these are homologous chromosomes. And just recap earlier, this just means that you have one paternal, this one is blue, and maternal and pink. They match up together, or they go and pair up. Let's just look at the definition from earlier I went over. So search homologous, go over top here. Right here. So a couple of homologous chromosomes or homologs are a set of one maternal and one paternal chromosome that pair up with each other inside a cell during fertilization. Yeah, so let's go back over to this now. So we go over here. All right, so this we have homologous chromosomes. Yeah, actually, I think it's pronounced homologous, not homologous. So homologous chromosomes, and they pair up before replication. And let's just keep reading. So following chromosomal DNA replication, the blue chromosome is composed of two identical sister chromatids, and the pink chromosome is composed of two identical sister chromatids. In mitosis, the sister chromatids separate into two daughter cells, but are now referred to as chromosomes rather than chromatids, much in the same way that one child is not referred to as a single twin. Yeah, so going back over here. Yeah, so we have these homologous chromosomes, which is pairing up paternal and maternal. chromosome, and then they each replicate separately. So then you have sister chromatids or twins. So you have twins here, sister of the paternal, sister of the maternal, and they switch over and they cross up, and now you have sister chromosomes in separate cells. And then, yeah, basically you're switching up each of these together. So you've just duplicated this setup right there. So that homologous chromosomes, now you separate up over here. And now you have two sets of those. And these are just called daughter cells. Or I mean, they're separated into two daughter cells, but now but these are the chromosomes in the daughter cells. But they're just referred to as chromosomes rather than chromatids. And yeah. And that's because they're not paired up with their twin, like this one over on top here, where you have the chromatids, the sister ones of those, and of each paternal and maternal. Yes, very fascinating. So let's just continue further. Now this is the cell cycle. So mitosis in an animal cycle, phases ordered counterclockwise. So it's going to go backwards is the order right there. So let's say... yeah, let's say we start over, let's just say over here, gap 1, cell grows. So this is cell grows, and then we're in the interphase stage where the DNA synthase or synthesis phase. Then this is the, yeah, this S phase is part of this interphase. So this is a subpart of it. You can see that earlier discussion here. Let's just go back over to that discussion. Let's move this over. Right here. Yeah, so in general, mitosis, division of the nucleus, is preceded, so it's preceded by S stage. So this S stage of interphase stage, during which a DNA is replicated, it precedes mitosis, and then followed by telophase and cytokinesis. Let's just keep going further here. So basically we have an interphase, and then this is the part of the interphase, which is the DNA synthesis phase. But the first part of that is actually this cell growing, this gap 1, G1, cell grows, and now we have DNA synthesis phase, S phase, called the S phase, cell duplicates its DNA, so, inside the nucleus, cell duplicates DNA, and you have centrosome duplicates, duplicates as well. And if you recall the centrosome, you have the centrioles and the microtubules inside there, so those get duplicated. That's providing the structure of the cell. And then there's a gap 2, G2 phase, or part of this synthesis phase. A cell grows some more. And let's read up on that gap 1 again, that we mentioned it earlier. Yeah, so the chromatode is 1/2 of duplicated chromosome during the synthesis S phase and occurring between gap 1 and gap 2 phases in the interphase portion of the cell cycle. Yeah, so it's gap 1 and gap 2 is the interphase, but the S phase is in between these two, and they're all part of the... the synthesis is the in between gap 1 and gap 2, and all these three. So gap 1, gap 2, and S phase are all inside the interphase portion of the cell cycle. Yeah, so these are all part of this right here, interphase, like that. All right, let's erase that. And yeah, continue here. So cell grows some more. So there's duplicated DNA and the centrosome duplicates. And now what we have is cell grows some more. And here's nucleus, the nuclear membrane, chromatin, and nucleolus. A nucleolus is the center one inside the nucleus. And here I quickly teleported to our earlier definition. So nucleolus. Let's try to pronounce that one more. Nucleolus. Yeah, nucleolus. Yeah, or nucleolus. Yeah, I think that's how to pronounce it. Just pause the video and Google search pronunciation. Remember, this is the largest structure in the nucleus, and it produces ribosomes, and that's inside the nucleus, like that. Anyways, continue back. All right, so we are back now, and yeah, so then that's the inside of the nucleus. The nucleolus is that little one inside there, produces ribosomes. Continue further. Now we're into prophase. Now this is the chromatin condenses into chromosomes. And now, let's just read up on chromatin. I haven't actually read up on it up to this part of the video. It was supposed to be later, but I think we should go over it now. So going back here, so there's a chromatin inside over there, and there's a nucleolus, and then there's nuclear membrane around. And then what happens is in the prophase, chromatin condenses into chromosomes, and nucleolus disappears. So that this one is gone now, which produces ribosomes. So that's gone. And let's go over here. So chromatin is a complex of DNA and protein found in eukaryotic cells. The primary function is to package long DNA molecules into more compact, denser structures. This prevents the strands from becoming tangled and also plays important roles in reinforcing the DNA during cell division and preventing DNA damage and regulating gene expression, which is the process of getting from DNA to RNA to proteins and so on. And also regulating DNA replication. So continue further just to illustrate this chromatin structure. So chromatin structure looks like this. You have a bunch of these core histones, H2B, H2A, etc. These are just different proteins. We'll get them over here. So histones are proteins that act as spools around which DNA binds to create structural units called nucleosomes. Going further right here. Yeah, so this is basic units of a chromatin structure. And what you'll have is core histones, core proteins over there. And then you have a histone octamer, which is a combination of all these. There's 8 of these. And then there is some DNA right here, 147 BP or base pair DNA. And then you'll wrap this around like a spool. So you'll form pretty much wrap around like a string on a spool where you're doing knitting and so on. So yeah, you wrap around these proteins right there. And this is beads on a string nucleosome or a nucleosome array. So basically, you have the histone proteins. You spool them up, and then you have it forms a structure called a structural units called nucleosomes. So you have nucleosome over there, or each one of these is a nucleosome. And then you form a whole bunch of these together. You pretty much have a chromatin structure. And more info on nucleosomes. A nucleosome is a basic structural unit. Let's try that again. Structural unit of DNA packaging in eukaryotes. The structure of a nucleosome consists of a segment of DNA wound around 8 histone proteins and resembles thread wrapped around a spool. The nucleosome is the fundamental subunit of chromatin. Each nucleosome is composed of a little less than 2 turns of DNA wrapped around a set of 8 proteins called histones, which are known as a histone octamer. There's 8 proteins right there. So octa is for 8, such as an octagon and so on. And wraps around almost about 2 full loops there. So a little less than 2 turns of DNA wrapped around a set of 8 proteins called histones, which are known as a histone octamer. Each histone octamer is composed of 2 copies of each of the histone proteins, H2A, H2B, H3, and H4. So you have 2 of these copies right there. H2B, you have H2B, H2A, then you have a second copy there, and there's an H3, H4. And then you have 2 copies of each of those, and those are right here. H2A, 2 of those. H2B, H3, H4, and so times by 2, that's 8. So going back over here, so there's a chromatin, which is pretty much that's a whole structure of wound DNA around proteins. And then what happens is the chromatin condenses, the proteins go away out of the structure, and then into chromosomes, and then nucleolus disappears. And now we're inside the mitotic phase, or the M phase. This is part of this one is a pro-metaphase. Nuclear membrane breaks down. So nuclear membrane, breaks down. Kinetochore or a kinetochore microtubules over there, and they invade the nuclear space and attach to kinetochores. And the kinetochores are I'll go over here actually. We'll go over it later, but it's a multi-protein complex. So it's via a multi-protein complex called the kinetochore. So anyways, going over there. So these microtubules attached to it. It's a bunch of protein there, multi-protein complex. And then what we'll have is polar microtubules push against each other, moving centrosomes apart. And remember, the centrosomes are the centrioles and microtubule structures. So now you have polar ones all pushing apart of each other, and then you have the metaphase. Chromosomes line up along metaphase plate. And here what I've done is just zoom in even more to see the better view right here. Chromosomes line up along metaphase plate, which is imaginary plane across here, which is in the center of these centrosomes being pulling apart everything. Then you have anaphase. Chromosomes break at centromeres, and the centromeres is where it's connected to it. Let's go back to that centromere. So that's the right here. Yeah, so with both copies, the centromere is a specialized DNA sequence of a chromosome that links up a pair of chromosomes. Sister chromatids called a dyad. Yeah, so that's where they're connected to it, and again, that's a specialized DNA sequence of a chromosome that links a pair of sister chromatids. Going back over here, and now you separate them across the centromeres, and sister chromatids move to opposite ends of the cell, so it breaks apart. Those are the sister chromatids, and these are the centrosomes pulling it apart. And then you have the telophase and cytokinesis phase. So nuclear membrane reforms, and nuclei reappear. Chromosomes unwind into chromatin. Yeah, this is the unwind into the chromatin setup, with that structure where there's a protein inside, but this unwinds, I don't like this word. I think it's better to say relax. So relax into chromatin. Yeah, so they relax into chromatin. Yeah, because you're basically being pulled apart, and you can consider that as winding. But actually, yeah, because there's no protein there, and then you could view the chromosomes as pretty much condensed, contracted with no proteins inside, and then they unwind by opening up, and then you'll have the chromatin. It then forms a chromatin structure where you'll have it like this. Yeah, so basically, imagine this was really condensed, and then it's unwinding itself, being more loose, and then to allowing the proteins to come inside, and then it could spool around it. Yeah, so we're back here. So now you have the nucleolus. The nucleolus is over here inside there. You can see that the nucleoli reappear inside both of these. So each one of these nucleolus, and the chromosomes unwind into chromatin, and nuclear membrane forms around. Then you have the myosin 2 and actin filament ring contract to cleave cell into two. So here's a cleavage furrow, and the word furrow is just a groove. Yeah, just think of it as a groove, or just a wedge in between. So cleavage furrow, or a groove inside here, and there's a contractile ring, that ring structure, and it will cut up. And it cuts up the two cells to form two separate cells right there as they're splitting up there. And this myosin 2 and this actin, let's just go over myosin, and what I'm going to do is actually just zoom out, and I also added a link here to the figure for the high definition zoom in, so we'll just have a better look at it soon. I'm just going to zoom out, get back to our previous size, like here. Alright, so we're here, and then so myosin 2, go over myosin. So myosins are a superfamily of motor proteins, best known for their roles in muscle contraction and in a wide range of other motility processes in eukaryotes. And motor proteins are a class of molecular motors that can move along the cytoplasm of animal cells. And we'll go over this in a bit, but motility right here is just the ability of an organism to move independently, and then while using metabolic energy. And now this is a very interesting aspect here. Motor proteins are a class of molecular motors. So molecular motors are natural or biological or artificial molecular machines that are essential agents of movement in living organisms. In general, a motor is a device that consumes energy in one form and converts it into motion or mechanical work. For example, many protein-based molecular motors harness the chemical free energy released by the hydrolysis or the water breaking reaction of ATP. And here we could just go and click over here just to get the definition right here. So remember, hydrolysis is water to unbind is any chemical reaction in which a molecule of water breaks one or more chemical bonds. So that's fascinating stuff there. Yeah, so it released by the hydrolysis of ATP, the energy currency, in order to perform mechanical work. In terms of energetic efficiency, this type of motor can be superior to currently available man-made motors. One important difference between molecular motors and macroscopic motors or large motors is that molecular motors operate in the thermal bath, which is an environment in which the fluctuations due to thermal noise are significant. Basically, a lot of motion or microscopic motion due to the movement of all the particles and so on, which correlates to the heat or the thermal or / kinetic energy. Now here's an example. Here's an animation right here of a ribosome, and it is a biological machine that utilizes protein dynamics. So notice right here, it takes the mRNA and translates it. So this is a ribosome, and it translates it to a specific set of amino acids to create a protein or other peptide and so on. So it has a whole chunk right here. And these are protein dynamics. It's just basically a bunch of movement of proteins, and then once it grabs it, it tries to follow the exact order encoded in this mRNA to produce this specific sequence there. It's fascinating stuff. So in proteins, to discuss on this protein dynamics, proteins are generally thought to adopt unique structures determined by their amino acid sequences. However, proteins are not strictly static objects, but rather populate ensembles of sometimes similar conformations. Yes, fascinating stuff. So let's go back over here. Yeah, so we have over here, and they split up into two right here, and then the last one here, resting phase, or GU, G subscript 0 phase. A cell leaves the cycle. Yeah, so one of the cells leaves the cycles. You have two, and now you have this other one that can keep duplicating itself, and the other one can duplicate itself on another separate cycle. And now here, this figure right here goes over to the cell cycle Wikipedia image. I'm gonna, that link goes actually not this one, but it goes to the actual link over here. So if you click this one, and then if you click again, so this cell cycle, so it goes to this image right there. That's Wikimedia. It's another Wikipedia Foundation website. So Wikimedia right there to basically stores a bunch of media and so on. So here's a bunch of here's that same one, but you could zoom in extremely close just to go over it all in detail. So you have this interphase, or we have this G1 cell grows, interphase, and then a DNA synthesis, S phase, cell duplicates its DNA, and we could even zoom in more, like that. So yes, fascinating stuff like that. So it's duplicating its DNA, and there is the centrosomes right there, also duplicates. So centrosomes duplicates, and then next one, let's zoom out even a bit more. The next phase, G2 gap 2 phase, cell grows some more, and then we have there's the zoom in to the chromatin and the nucleolus in the cell membrane, and there's the chromosomes inside, and then let's see this phase right here, the prophase. Chromatin condenses into chromosomes. Yeah, so there's condensed all those loops into chromosomes. They get so condensed. They get really small because there's no more protein histones inside. And here's some astral microtubules. These are just far distant ones away from these. These ones are connected over to the chromosomes. And then these set up right here. Yeah, so these microtubules invade the nuclear space and attach to these other... these kinetochores. Let's go back to this here. So remember, these are just a multi-protein complex called a kinetochore. And we'll go over those in a bit in more detail in this zoom-in image of this one. Going back here. So yeah, so this attaches over to it. Yeah, which is utilized to connect to the chromosomes, and then there's polar microtubules push against each other, moving centrosomes apart. So it moves all these apart and separates all over. And then this is the mitotic phase or M phase going over here. Zoom back a bit. So now here we have the metaphase. Chromosomes line up. Metaphase plate, imaginary plate is right over here. And then there's another zoom in of the chromosome. And there's the centromere, which is that binding spot right of these two chromatids. And yeah, these are two sister chromatids right there. And there's this is a kinetochore or kinetochore right there, which is attached to this chromosome that can attach to these microtubules. And then the next phase, the anaphase, the anaphase right here. Chromosomes break at centromeres, and sister chromatids move to opposite ends of the cell. Those are sister chromatids. Right, like there, and then we have over here the telophase and cytokinesis. And what we have is so the nuclear membrane reforms on both sides, and nuclei reappear inside. So there's a nucleus. There's a nucleoli, so the inner nucleolus of each one, and then chromosomes unwind into chromatids because they're condensed, and they unwind, and then they allow the histones to attach and then get spool and then spool around those. And myosin 2 and actin filament ring contract to cleave cell in two. And remember what actin was. Let's go back to refresher. Actin, we mentioned this earlier. Let's do a space actin. And yeah, so actin is a family of globular or spherical multifunctional proteins that form microfilaments in the cytoskeleton, and the thin filaments in muscle fibrils, yeah, or fibrils, which are rod-like organelle of a muscle cell. It can be present as either a free monomer called G-actin globular or as part of a linear polymer microfilament called F-actin. And this is filamentous, or just filaments, and so on. Alright, so let's get back over here. Yeah, so this splits it up, and then we have these separate over there into two. And you also have, and then you have two sets of these centrosomes where the centrioles and so on are inside. And the next one is resting phase. G0 cell leaves. So one cell leaves, and now you have another one that can keep replicating this cycle. Hence, the word cycle keeps going. So this is for animal cells. This fascinating stuff. Let's get back into our rhythm right here. Move this over. All right. So now continue further, and let's look at this one. Here's a close-up of mitosis, which again divides the chromosomes in a cell nucleus. So you have DNA replication. And notice how these chromosomes are replicated, or the DNA is replicated. So this one right here replicates, and then it has this centromere connected at the center there. And there's the chromatids, sister chromatids of each of these paternal and maternal. And then you'll have the mitosis, which is the division of the nuclei. So basically, you'll have this, and it divides into two, and divides across these across these sister chromatids of both of these pairs, and you'll have one pair of each, or one chromosome of each, and then you'll have these two diploid cells, which have two sets of the chromosomes like that. And so one is from each of the parent. And so fascinating. So let's keep going further. So that's mitosis divides the chromosomes in a cell nucleus. And going further, the spindle apparatus. Yeah, this is the part where it's these microtubules that's pulling apart on the chromosomes right there. So the spindle apparatus, or mitotic spindle, refers to the cytoskeletal structure of eukaryotic cells that forms during cell division to separate sister chromatids between daughter cells. So it connects over to here. And this is the kinetochore, the proteins there. It connects to them, and this is the kinetochore microtubules, the ones that are connected on it. And this is K fibers. And astral microtubules are the ones not connected to this right here. So notice how it's darkened in green, just to illustrate the difference. And there's the microtubule organizing center, MTOC. So let's go over here first. This diagram depicts the organization of a typical mitotic spindle or spindle apparatus found in animal cells. Chromosomes are attached to kinetochore microtubules via a multi-protein complex called the kinetochore. And that's right on it there. Let's make this a bit smaller. And these are polar microtubules that want to separate the centrosomes apart. And there's the MTOC, the microtubule organizing center. And then the microtubules that aren't connected to a kinetochore are called astral microtubules, separated like that. So fascinating stuff here. Now here is a micrograph image of it. So and this is a micrograph showing condensed chromosomes in blue. So this is these are the condensed chromosomes, so they're not inside their spools or the histones anymore. And then there's the kinetochores in pink. So those dots are on the chromosomes there that basically can attach to these microtubules. And microtubules in green during metaphase of mitosis. And let's go over that metaphase again in the big diagram. The metaphase right here, chromosomes line up along metaphase plate, which is an imaginary plane right over here. Yeah, just basically in between the two centrosomes, and then they get pulled apart. Fascinating stuff. All right, let's continue further. So now here's another image of human chromosomes during metaphase, and where they're lining up, and then they get split apart. Yeah, so they're all bunched up together there and connected at the centromere, or the center part where it's connected, or just the connected part of the two chromatids. Continue further. Now here's another image here. Basically diving deep into this one. This is very important because pretty much all of biology stems from the cell division. or at least the theory of this cell division, how it's done, and so on. So stages of early mitosis in a vertebrate cell with micrographs of chromatids. All right, so here's the stages of it. Let's look at the prophase. Yeah, so we have the prophase where we have the this is the kinetochore is attached. These are the sequence on the two chromatids, and these are sister chromatids, and they're condensing, so they're removing the histones, and they're basically so now they're not spooled up anymore, and they can condense in a very small package. And then you have the mitotic spindle forming, or the spindle apparatus, and then these are the it's over here, nuclear envelope around the nucleus right there. And there's a centrosome right here, which has a microtubules extending out. And there is a micrograph of the prophase. There's the two chromatids. Yes, very interesting. So now continue further. Yeah, so this is more condensed in. Now there's the metaphase. So we have fragments of nuclear envelope over here, because the nuclear envelope is going to be separated apart. Let's just zoom into this. I'm going to zoom in more. All right, so we're zooming in. All right, yeah, so zoomed in here. Yeah, so there's prometaphase. There's the fragments of nuclear envelope that breaks apart. And then you have the centrosomes over there. And now they're connecting over to the kinetochore, which is on the chromosomes, or on the chromatids. And then there's a prometaphase right there. They're connected. Notice how they're very bunched up together like that. And then there's the metaphase, where they're all lining up around this imaginary plane. So there's the kinetochore microtubule, which is connected to it. And then the astral microtubules are the ones not connected to the kinetochore. And there is metaphase right here, so they're all lined up, zooming into it, so it's pretty much lined up on an imaginary plane down the middle. And there's the anaphase, and this is where they separate apart. So kinetochore microtubule shortening by pulling it apart. It was pulling it apart and shortening, and now these are getting separated. Here's early anaphase there, and just before it's fully separated. All right, let's just zoom out and continue further. Some fascinating stuff. And now here is an animation of this. So animation, label-free live cell imaging of mesenchymal, as to Google the pronunciation. And then this is, which is connective tissue, stem cells, which can change and differentiate into another cell type. We'll get over those later. And then undergoing mitosis. And yes, this is a fast animation there. Notice, let's see how it is. Yeah, so basically you have the cell nucleus, the nuclear envelope separates, and then you have over here the chromatids all separating on this imaginary line. Let's try to pause it. Yeah, let's just pause here. Okay, here's initially, so you have a bunch of stuff there. There's the chromatids all connecting. It's hard to see, but the idea is there. And then you have them, let's just keep going. And then stop it. Yeah, so now you have it lining up, and then it's going to start getting pulled across. Okay, so there it is, and let's see. Okay, so it always goes backwards. Okay, and it's going to start being pulled apart right over here. Yeah, and then it's pulled apart. Fascinating stuff. So it's very fascinating. And note here, most human cells are produced by mitotic cell division. Important exceptions include the gametes, which are the sperm and egg cells, which are produced by meiosis. Yeah, so the mitotic cell division, it just replicates itself. But the special ones here, the gamete, sperm, and egg cells, such as these ones, are produced by meiosis, and we'll go over that now. So, in contrast to mitosis, meiosis results in 4 haploid daughter cells by undergoing 1 round of DNA replication, followed by 2 divisions, or 2 mitosis stage. No, actually, no, it's not 2 mitosis. It's going to be 2 meiosis stages there. So, going over here. So, basically, 4 haploid, which remember, these are half of the number of sets of chromosomes, as opposed to having a diploid and then over to a diploid. This is going to be a haploid. Yes, so it's going to be half. So if you have a diploid, it's going to get 4 half of the chromosome set daughter cells, or haploid cells. So let's continue further. So it's going to so meiosis results in 4 haploid daughter cells by undergoing 1 round of DNA replication, followed by 2 divisions. So homologous chromosomes, which are the parental chromosomes lining up, are separated in the first division, meiosis 1, and sister chromatids are separated in the second division, meiosis 2. Yes, and I'll show those soon. So both of these cell division cycles are used in the process of sexual reproduction at some point in their life cycle, Both are believed to be present in the last eukaryotic common ancestor. That's fascinating. Okay, now let's continue further. So here's an image here. Let's just make this a bit zoom in to get the text there. All right, so in meiosis, the chromosomes duplicate. So we have here chromosomes lined up. These are the homologous chromosomes. Yeah, the homologous chromosomes are right here. They're going to be lining up. They exchange genetic information during meiosis 1, and then the daughter cells divide again in meiosis 2 to form haploid gametes. So let's look at this earlier again. So basically, yeah, we have the first part right here, the interphase. The chromosomes duplicate. I think actually interphase is over from, yeah, this is the interphase. It duplicates there. Yeah, so there's a duplicate. So there's the chromosomes right there of each parent. So let's say this blue is the paternal, this is the maternal, is the red, and then it duplicates and it connected at the centromere, or just a central locus there, or just a locus point that connects it. So it duplicates there. So this becomes this, and then this big one becomes. Let's move this over. Yeah, so I'm going into detail on this. It's also a learning experience for me. This goes into this, and then you have this big one here goes over to here, and then these same with the red ones. So it duplicates, and then what ends up happening is then they pair up together. So you have a paternal, maternal. So homologous chromosomes pair up, and these ones pair up. So these are the like ones, and then what we'll have is these exchange. So this is the first meiosis 1. So the homologous chromosomes exchange genetic information during meiosis 1. Yeah, so let's look at this diagram right here in meiosis 1. So we have this right here, which has, this is the big pair of chromosomes, and then has one of these ones there. So then this goes over here. And likewise over here on this side, we have this part. Yeah, actually, these are just interchanged. Yeah, so this is what ends up happening is this part here, they exchange genetic information. So this goes to here, and they replace each other. Actually, to line up better, this one is over here. This one. So we have this pair, and this part goes over to this part. So then this one's going to be blue, so it's going to match up here. And this one's going to be red, which is going to match up here. And likewise on this little chromosome set, or chromosome pair. So what we'll have is this one becomes red, so that this means this gets exchanged, and then this gets exchanged with this part right here. So these interchange like that, and they interchange. This one becomes red there, which is this. And then this one, then this one becomes blue there, which becomes this. So that's fascinating stuff. So they exchange there in meiosis 1, and then there's a daughter nuclei, and it splits up as well. Yes, you have two cell divisions. So these intermix, and then they separate. Now you have these, and notice that these ones are haploid. These are diploid because that's two sets of each of these chromosomes, or two pairs of each one. I mean, so as a pair of each one. And then what we'll do is the next is meiosis 2, and then daughter cells divide again into meiosis 2 to form haploid gametes. Yeah, which are the sperm or egg cells, like that. And then these split up again, and then what you end up with is, well, this part's going to split up, and then it's going to go with this part, and so on. So you have it over there. And then you also have this part is going to split up. Let's say this part is going to split up, and it's going to go with this one here. That's going to go over here. And then the next one is this one goes with this one. Yeah, so these just split up accordingly. So that's fascinating stuff. And now you have 4 of these. And then these are all haploid, because these are each have one set. So one here, and then there's one here. This one has two. So it goes from diploid to haploid there. That's fascinating stuff. Let's just keep going further, and just to show the difference between meiosis and mitosis. So figure comparison of a meiotic meiotic and mitotic processes. So we have meiosis makes distinct gametes, so diploid to haploid. So what you end up having is, yes, 2 cell divisions here. So these first they duplicate and exchange the genetic material. Notice how there's different kinds there and there. And then they split up. They split up into 2 of these, into 2 daughter cells. And then these are diploid, and it goes from diploid now, and it splits up to haploid. So it splits up again. So that is meiosis. And then mitosis is, let me just erase this. So mitosis clones body cells diploid to diploid. So you have, here's DNA replication of the first was diploid at 2 sets. It's 2 pairs of chromosomes. Or each set has 2, has a pair. And then it splits up, and then you still have these diploid right there. Yeah, so it divides up, and then you still have over here. Because remember, at first you duplicate and then divide. So then you're going to still have these 2 pairs. So this one's a pair, and these ones are paired up as well. And this is a pair. You get this one together, and there's the pair over there. Whereas over here, they're all haploids, they're all just 1, 1, and 1. This one is 1, 1. And then over here is the second. Yeah, this one's also 1, 1. These connect to each other. And the other ones are the set of that. Just fascinating. Let's continue further. Now here's another table to illustrate the differences further. So figure meiotic versus mitotic processes. So here's mitosis. Division's 1. Meiosis is 2. So here's a full comparison in more detail with the table. Let's erase this. Yeah, and then the next one, independent assortment, mitosis no, and meiosis yes. Metaphase 1. Let's look at that metaphase 1. It's when they line up together, and I'll get to that actually in a bit. But before we get to that, this independent assortment, let's just read up on this one. And yeah, I had to pause the video and add this actually extra stuff. So law of independent assortment. This is from figures, actually from YouTube video. So allele is a gene variant, and we'll get over to that in a bit. So law of independent assortment. During gamete formation, different pairs of alleles segregate independently of each other. And then you'll have, for example, you have a parent cell right here. So you have A and B. Which allele A, which is a gene variant, a gamete receives. So which allele a gamete receives for gene A, so gene A, has no bearing on the allele it receives for gene B. So if gene A, and there's gene B, and then there's little b, and then so basically produced evenly in meiosis. Gametes are produced evenly. So which one it gets? Yeah, which one it gets? For example, here, this A right here, I'll make this in a white one. Yeah, let's just click this draw button. And I'm going to change this into white. Yeah, so basically, which one here, so this A. So this A, if it receives B, this capital B, if it goes over to there, it does not depend on what this one was getting. It does not depend on what this one, if this one gets A, it does not matter. So it's always going to be evenly produced. It produced evenly A, so A will have this capital B, A will have this lowercase b, and then this B will have A, and then this other one here will be produced together. Yeah, which is this A, lowercase a, and lowercase b there. So they just produce those. Let's erase this and change this back into black. All right, continue further. All right, so continue further. So a gene, just to recap, or just to get caught up on this, I'll go over later on genes and gene variations or alleles later in this video, but I will go over some right now. So a gene, from the word from the Greek genos, meaning generation or birth or gender, is a basic unit of heredity and a sequence of nucleotides and DNA that encodes the synthesis of a gene product, either RNA or protein. So if you have a chromosome like here, this is 10 to the 7, or 10 to the 7 to 10 to the 10 base pairs. Yeah, so that's in the tens of millions to over a billion base pairs, because 10 to the 9 is a billion. So that's in the billions, millions and billions. And then so you have the chromosomes right there, two pairs, or this is 2 sets of it connected at this locus here. And then there's a DNA strand, just to zoom in there. And there's the gene. It could be a set amount of base pairs. So for example, so one gene 10 to the 3 base pairs, or to 10 to the 6 base pairs. So it could be this one could be 1,000 to a million base pair length. So a gene is a region of DNA that encodes a function. A chromosome consists of a long strand of DNA containing many genes. A human chromosome can have up to 500 million base pairs. And base pairs of DNA with thousands of genes. Fascinating. And the term allele, modern formation from Greek allos or other, denotes the variant of a given gene. In genetics, it is normal for genes to show deviations or diversity, and all alleles together make up the set of genetic information that defines a gene. So yes, if it's producing a specific function or variation of that function, and you'll have at that specific spot, it can have a variation there of that gene, and then basically all the different variations of that specific type of gene is considered a gene. And going over chromosomal crossover, that's the next one. So mitosis generally does not have independent assortment, but in some cases it may have it, but not always a good thing. So here's meiosis, so it does generally. Yeah, so the meiosis does have it. And then synapsis. Yeah, this synapsis is when they line up, when the pairs of chromosomes line up together. Yeah, so these ones would line up, and this one would line up, and we'll illustrate that here. Let's just go over that a bit here. So synapsis is the pairing of two chromosomes that occurs during meiosis. It allows matching up of homologous pairs prior to their segregation and possible chromosomal crossover between them. And yeah, let's read up more on this and just show a figure here. Here's synapsis during meiosis. The circled area is the part where synapsis occurs, where the two chromatids meet before crossing over. So here's meiosis. Here, two homologous chromosomes from the paternal and maternal, and now they line up together. You know, they pair up or meet up. And then what happens crossover is when they have the genetic information interchange right here. So this one right here switches up with this, like that, and that is crossover. So let's read up on that. Chromosomal crossover or crossing over is the exchange of genetic material during sexual reproduction between two homologous chromosomes, non-sister chromatids, that result in recombinant chromosomes. Yeah, so these chromosomes, they're interchanging with a non-sister. So these ones are sisters, and these ones aren't. Yeah, this one is a non-sister relative to these two, relative to each other. And then they meet up and swap up genetic information, and now you have recombinant chromosomes, or which is a combination of two others. Going further, and now here's a further diagram. So crossing over occurs between prophase 1 and metaphase 1 the phases, and is the process where two homologous non-sister chromatids pair up with each other and exchange different segments of genetic material to form two recombinant chromosome sister chromatids. So here's an example right here. So this one lined up right here. So look at this gene or a location of a sequence, A, capital A, and then this one's lowercase b. And this one has A and lowercase b. And these are just duplicates of each other. And notice this one here is lowercase a, capital B. And then what happens is this would swap up with this. And then they get split up afterwards. So they get all split up. And what you end up with is you end up with right here. This one is just this one. This one is just this. It's over here. And then you end up with this part right here, which is that crossover one that's over here. And then we have the second crossover one here. And that is right here. And then the last one is by itself. That's over here. So yes, let's just erase all this again. So it's very interesting stuff. Let's continue further on this paragraph, just regarding this figure and crossing over in general. It can also happen during mitotic division. So it's very interesting right here. And here I just bolded that just because it's supposedly mitotic division or mitosis doesn't have that, but in special cases it does, and this which may result in loss of heterozygosity. Yeah, or heterozygosity, yeah, I think it's just something like that. Anyways, crossing over is important for the normal segregation of chromosomes during meiosis, and we'll get over this loss of heterozygosity. It basically means loss of genetic diversity. So crossing over is important for the normal segregation of chromosomes during meiosis. Crossing over also counts for genetic variation because due to the swapping of genetic material during crossover, the chromatids held together by the centromere are no longer identical. So when the chromosomes go on to meiosis 2 and separate, some of the daughter cells receive daughter chromosomes with recombinant alleles. Due to this genetic recombination, the offspring have a different set of alleles and genes than their parents do. In the diagram, genes B, capital B, and lowercase b are crossed over with each other, making the resulting recombinants after meiosis a capital A, lowercase b, then capitals AB, then lowercase ab, and then the other one is lowercase a, and then capital B. Yeah, and you can see those genes B, capital B's and lowercase b. That's just over here. So if you have right here, initially it's A and then this one here, and then it swaps over with this capital B. So then this becomes capital B, and then this one gets swapped over, crosses over to the lowercase b side. So yes, very interesting right there. And go over this loss of heterozygosity. So loss of heterozygosity, or LOH, is a cross-chromosomal event that results in loss of the entire gene and the surrounding chromosomal region. So you can have a crossover in mitotic division, in which may result in loss of heterozygosity. So one of them could be lost as opposed to even swapping. Yeah, so technically not the exact same as this crossing over or swapping over there. But yeah, just wanted to include that. This is just from Wikipedia, so it's very interesting. And yeah, so that's basically loss of genetic diversity when you have a loss of an entire gene. It could be a loss, yeah, I believe you could also be swapped with another gene that is a standard one. For example, here, a diploid organism is called heterozygous at a gene locus or specific region when its cells contain two different alleles or gene variants, one wild type, which is a typical form allele, and one mutant, which is a non-standard allele of a gene. Yeah, so then if you lose one of those genes, for example, if you lose the mutant one, you're going to be less diverse because now you're just getting more of these wild types, or the typical ones. And another set here, well, just another definition I want to go over is bivalent, and that's because it's included here. So mitosis, generally no, it doesn't have a synapsis, the lining up of the chromosome, the homologous chromosomes, but meiosis does, and then they form a bivalence. You go over a bivalent right here. Right, so a bivalent is one pair of chromosomes or sister chromatids in a tetrad. Yeah, tetrad. A tetrad is the association of a pair of homologous chromosomes, which is 4 sister chromatids, physically held together by at least one DNA crossover. And yeah, this physical attachment allows for alignment and segregation of the homologous chromosomes in the first meiotic division. And here's a diagram, basically just to simplify what these are, and its definitions. They're pretty much the same, almost interchangeably. So a bivalent is a pair of homologous chromosomes. So here's a pair of these two chromosomes, and then so this is one chromosome, and this is two. These are duplicates of each other, so it has each one has 2 chromatids. And then if you wanna, then you could also be calling it a tetrad, which is 4 homologous chromatids or 2 homologous chromosomes, this is basically a bivalent. So it depends on how you are, how you're counting them up. This could be bivalent. It's essentially the same thing, but this diagram, what it looks like here, over here, yeah, it looks like over here is before the crossover. This is after crossover, but then Wikipedia says it's held together by at least one DNA crossover, so it could be an interchangeable. And again, bivalent is looking at it as a pair of chromosomes, and a tetrad is looking at it as 4 chromatids. And essentially the same thing. I believe it has to do with the different phases of the cycle. When they're if they're very closely connected, you might not be able to see the chromatids that easily, or the separation won't be that easily. So you'll just see them as 2 chromosome pairs. And then when they're more separated, you can see that you may be calling it a tetrad. I believe that's the reasoning behind it. Anyways, let's get back into business here. I want to go back to this table, and again, yeah, diving deep into this just so it gets stuck in our brains. Okay, so now crossing over, mitosis doesn't have it, because it just duplicates, and then meiosis, yes, in prophase 1, where it exchanges the genetic material, and then the outcome is mitosis has 2 cells, meiosis has 4 cells. And then ploidy, this one is going to be just diploid. Yes, you're going to be the same number of chromosomes, go from 2, the same number of sets, so 2, and then it goes to you duplicate into 2 cells, each have 2. But meiosis has a haploid, so it has half of the chromosomes that you're duplicating. So if you go back over here, so you have this one has 2 sets, and then you're divided into 2. Now you have 1 set of each of these chromosomes. This one initially you had 2 sets. So you had 1 little one, 1 small one, 1 big one, and then another big one. So 2 big ones from maternal, paternal, and 2 small ones, maternal, paternal. And then the next one, the use for mitosis is body cells, so just to duplicate and create pretty much the entire body. And then the meiosis, the sex cells, or the gametes, the sperm and egg. And then genetics, identical cells, and then meiosis is variation. That's fascinating. And now let's go over further. And here's another comparison diagram. This one here is more detail. So this figure sourced from over here. Tortura, or Torto, I'm not sure exactly how to pronounce that. And then this is Principles of Anatomy, Physiology, 12th edition, 2008, right there. And let's just look at it over here. Yeah, and I got this figure from, let's just hover it over, so biology.stackexchange.com. And let's just do zoom in here, zoom in closer, and center this. All right, so compare this mitosis in the blue region and then the meiosis in this yellowish region. So you have the starting cell right here, and let's say it has 2N, or chromosomes are already duplicated inside the set. Yeah, so they both duplicate the chromosomes right here. Yeah, so this 2N is a diploid cell, so it has 2 sets of N number of chromosomes, and then so these are already duplicated, and then so that's a starting cell. Then in meiosis, what happens is we'll have this crossing over, this prophase 1 crossing over, and then tetrads formed by synapsis, so they line up, and then there's the crossing over. And then in metaphase 1, tetrads line up along the metaphase plate, and then they line up, and then anaphase 1, homologous chromosomes separate, sister chromatids remain together. Yeah, so they remain together, but yeah, so these line up, and then they remain together. It's the sister ones, but the homologous chromosomes separate. Then telophase 1, each cell has one of the replicated chromosomes from each homologous pair of chromosomes N. Yeah, so this would be a haploid at this section, because notice here, this one here has 2 sets of each one. They line up. Now you have 1 set. So you have 2N, and then you have 1N. And then, yeah, they go over here. So each cell has one of the replicated chromosomes from each homologous pair of chromosomes. And then that's the prophase 1 for meiosis. But in the prophase over here for the mitosis, here I quickly just teleport above to the other image. The prophase, we have this section here where the spools of chromosomes, they release from those histones, and then they're by themselves. So you could see it above here, and I just teleported above here. So prophase chromatin condenses into chromosomes. Remember, the chromatin is the spooled up chromosomes, and then it condenses into it. So it releases the histones, and it goes basically condenses super small. So here's that DNA strand, and then it's around this over here, spooled up. And then when you remove these, then all you get is this here, and it condenses very small, because these are still adding a bunch of volume there with the proteins. Anyway, let's go back to our illustration. All right, so we are over here, and we're zoomed in a lot. And let's look at it. Okay, so now we're over here. That's prophase there. So there is the chromatins turn into or condense into the chromosomes. And then, okay, so here's prophase 2 for the meiosis stage. And again, this lining up. And let's see what's the next section here. Metaphase 2. This one's metaphase, just normal one for mitosis. They all line up. Chromosomes line up along the metaphase plate. So they line up again. And this prophase 2, they split up. So notice how they're split up from this right here, and they go over here. And they split up. And then now what we have is lining up. All right. And then they line up around this plate or imaginary line there. And then that's the anaphase. This is anaphase 2, because it's the second one. It's already done that over here. It's already lined up and split up. It's anaphase 1. No, that's metaphase. So this metaphase 1 is lining up. Metaphase 1 is lining up. And this is prophase. Here's a crossover. This one is just a condenser. And then the next one here is prophase 2. This one is prophase. Yeah, they're just lining up now. Lining up together. Or they link up with each other. Yeah, so they link up, and then this one is lining up in the imaginary plate. And anaphase 2 is they pull apart. Sister chromatids separate. Anaphase just note 1 for the mitosis. And then cytokinesis. Yeah, this cytokinesis is when they split in half and get their 2 daughter cells. So each one of these has a cytokinesis here, and this is in the telophase 2. So remember, telophase 1, there's the replicated chromosomes, and they split up there. in half. And I'll just write down here. So I just put here cytokinesis is the part of the cell division process during which the cytoplasm of a single eukaryotic cell divides into 2 daughter cells. So yes, this one divides into 2 right there. And then notice the diploidy. This one has N, and then these are all N. So basically, one set of the chromosome of each chromosome. This one has 2 N, so notice it duplicates across there in the mitosis. Mitosis just duplicates itself. Yeah, so just duplicates. This one is gets 4 daughter cells, and it has half the diploidy, or the number of sets of chromosome. That's fascinating stuff. So now let's zoom out. Alright, yeah, so now let's look at the next section. This is on prokaryotes, and I just want to go scroll all the way back up before we started in this deep dive in cell cycles and mitosis versus meiosis, and this is for eukaryotes, so basically organisms with cells that have a nucleus in each one, and the other ones, prokaryotes don't have one. Yeah, they don't have a nucleus. So let's just keep going up just to show where I left off, where we said it was. Yeah, so this whole cell cycle one was, the tangent that I went off on was in eukaryotes. There's 2 distinct types of cell division, mitosis and meiosis. And what I'm going to do is I'm going to put enter like this, just to separate it. So this is eukaryotes, and then the next one's going to be on prokaryotes. And let's scroll all the way over to it. And that's going to be right here. Alright. So prokaryotes, i.e. archaea, this is just simpler and evolutionarily older than bacteria. So very, very similar to it, and bacteria. So these are 2 types of eukaryotes. So just for example, can also undergo cell division or binary fission, division into 2 parts. Unlike the processes of mitosis and meiosis in eukaryotes, binary fission takes place. Actually, I think this is a typo by Wikipedia because I copied and pasted it to seek. Let's put that so you know it's not from me. So, so unlike the processes of mitosis and meiosis in eukaryotes, binary fission in, I'm just going to skip that text. So binary fission in prokaryotes takes place without the formation of a spindle apparatus on the cell. Remember, the spindle apparatus is this setup here where it's pulling apart the chromosomes. So there's no need for the spindle apparatus. And so before binary fission, DNA in the bacterium is tightly coiled. After it has uncoiled and duplicated, it is pulled to the separate poles of the bacterium as it increases the size to prepare for splitting. Growth of a new cell wall begins to separate the bacterium, triggered by FTSZ polymerization and, quote, Z-ring formation. Alright, so now let's go over this FTSZ. Let's note FTSZ stands for filamenting temperature-sensitive mutant Z, is a protein that assembles into a ring at the future site of bacterial cell division, also called the cell ring. The Z ring. So here is a example of it. So the Z ring forms from smaller subunits of FTSZ filaments. So as a protein, it's a bunch of proteins, or subunits of that protein or type of protein. These filaments may pull on each other and tighten to divide the cell. So you have a whole bunch around there, and they can pull on each other to tighten at a specific place. Because if you imagine this in 3D, and we'll cut it, and you could split it in half. All right, so here is another image right here. This is a super resolution image of Z rings green at different stages of constriction in 2 E. coli cells, a type of bacteria. And the super resolution is a type of imaging that enhances the resolution of types of imaging technology. But yeah, that's just a bit beyond this video. Let's just keep going further. So binary fission, here's just a figure of it. Here's a simple diagram. So what you have is, here is the cell envelope or cell membrane, and there's the DNA right there. Then there's the FTSZ ring, that protein one, that can constrict or pull together and then constrict and divide a cell. So these are Z ring forms from small units of the FTSZ filaments, and they can pull upon each other. So this is assembles into a ring at the future site of bacterial cell division, also called the Z ring. So this could form around this ring right there, and then it can all pull itself towards each other, and then just split this in half. And now you have two DNAs and 2 cells. Yeah, so split up. And here is a figure S aureus cell, or cells a type of again bacteria, or just generally archaea. So yes, you have this, and there's a Z ring, and it splits it in half. And the new cell wall, the cell envelope, just basically cell membrane and wall or I believe. In the new cell wall called septum fully develops, resulting in the complete split of the bacterium. The new daughter cells have tightly coiled DNA rods, ribosomes, and plasmids. Note that a plasmid is a small extrachromosomal DNA molecule with a cell that is physically separated from chromosomal DNA. And can replicate independently, so it's not in the chromosomes. So outside of that, in nature, plasmids often carry genes that benefit the survival of the organism and confer or give to a selective advantage, such as antibiotic resistance. And extrachromosomal DNA, abbreviated ecDNA, is any DNA that is found off the chromosomes, either inside or outside the nucleus of a cell. Most DNA in an individual genome is found in chromosomes contained in the nucleus. And this is for eukaryotes. And so going further, so antimicrobial resistance, AMR, occurs when microbes evolve mechanisms that protect them from the effects of antimicrobials. Going further, an antimicrobial is an agent that kills microorganisms or stops their growth, and antimicrobial medicines can be grouped according to the microorganisms they act primarily against. For example, antibiotics are used against bacteria, and antifungals are used against fungi. That's fascinating stuff, and we'll go over fungi slash fungus later in this video. Bacterial DNA. So here is the DNA in this one, illustration of bacterium showing chromosomal DNA and plasmids. Note that there's no nucleus in this bacterium. And you just note that bacterium is singular, so just representing one. Singular. Versus bacteria is plural. Yeah, so illustration of a bacterium showing chromosomal DNA and plasmids, not to scales, just to illustrate. So there is the bacterial DNA, and then you have some plasmids that are smaller ones that are apart from this longer strand right there. And here is an electron micrograph of a DNA fiber bundle, presumably of a single bacterial chromosome loop. So there's a big one right there that could be considered DNA. Then you have this little extrachromosomal DNA, or chromosomal DNA, that's the plasmid. It's fascinating stuff.